You don't need to be a full-time biotech analyst to get a sense for the burgeoning RNA therapeutics landscape. The space has been sprouting new company growth at a dizzying pace, particularly on the heels of the COVID pandemic, and it's showing no signs of slowing down. While the development of RNA therapeutics is arguably the brightest spot on the biotech horizon, you might also argue that the public and private research infrastructure required to support the advance of RNA as a therapeutic modality has some catching up to do. I'm Matt Pillar.
And I'm Anna Rose Welch.
And on today's episode of the Business of Biotech, we're going to take a deep dive with a couple of RNA industry notables who are working hard to create a world-class research and development resource for RNA innovators centered around a world-class resource of its own right at Johns Hopkins University's Institute for Nanobiotechnology. If you're into RNA, our guests likely need no introduction. Dr. Jeff Coller is a Bloomberg Distinguished Professor of RNA Biology and Therapeutics at Johns Hopkins University, and he serves on the boards of the Society for RNA Therapeutics and the Alliance for mRNA Medicines. His colleague, Dr. Michelle Kim, is managing director of the new Johns Hopkins RNA Innovation Center, the ambitious initiative the duo are spearheading. A Hopkins molecular biology and biochem PhD herself, Dr. Kim brings more than a decade of research experience earned at Howard Hughes Medical Institute, Circulomics, and PacBio to the party. Jeff and Michelle, welcome to the show.
Thank you. Thanks for having us.
Joining us. We're happy to have you.
Yeah, super happy to have you. Yeah, and
no, we're excited to be here.
Yeah, we had the benefit of a conversation a few weeks ago with Jeff to talk a little bit about what's going on there, and it's a relatively new initiative that we're excited to share with the audience because I think the work that you guys are doing is going to be awfully intriguing to the folks who are interested in RNA, who are Business Biotech listeners. And I thought,
which should be everyone.
Yeah, I mean,
should be everyone.
Yeah, you might not be an RNA developer, but everybody's RNA curious, right?
Should be.
They
have RNA all throughout them. So
yeah, everyone has RNA.
We are mRNA. Come on.
That's painting with a pretty broad brush.
On a t-shirt, right?
We'll go with it. We'll go with it.
All right.
I thought maybe we'd start with the origin story of the Johns Hopkins RNA Innovation Center. What was the sort of the nucleus of thought, that the kernel of thought that sprouted this initiative?
Yeah, it really started back during the pandemic, and so the origin story really goes way back to like 2012. I first started working on mRNA-based therapeutics back in that time, and the space of mRNA therapeutics was something that most people just weren't paying much attention to. And I had gone to some of these meetings that were very small at the time, only a couple dozen people, and had met some of the big players in that field. In particular, a guy named Mike Houston, who is a CSO of a very small company out of Seattle called PhaseRx. And I was talking with them, and Mike and I had some ideas about, how to improve mRNA-based therapeutics. This was more than 10, 15 years ago now. Mm-hmm. And I started working with PhaseRx, and sadly, they basically went under. But during the pandemic, Mike gave me a call, and he was now CSO of TriLink Biotechnologies, which, as we all know, bloomed into this huge organization during the pandemic because they were supplying CleanCap for Comirnaty, or Pfizer's version of the vaccine. And what was clear in some of the conversations that Mike and I had was there's a lot of interest in the RNA therapeutic space, but there's only a few dozen labs on the planet that are even working on mRNA translation, mRNA stability. And so how are you going to develop a new therapeutic modality, which has really become a pillar of medicine now, and innovate that if the academic support network just doesn't exist? And, one of the rate limiting steps, of course, with getting academics interested in this is feasibility and accessibility to materials at a reduced cost, and at a cost point where they can do rapid experiments and try to innovate around the iterative experiments that they would do. And Mike and I started talking about this, and he put me together with the one of the founders of Maravai, which is the parent company of TriLink, a guy named Carl Hall. And Carl and I had a conversation around this where we thought, wouldn't it be great if we could get into academics' hands at a very reduced cost RNA-based molecules that they could then, rapidly screen through, rapidly innovate their technology around, and then do experimentations and continue to evolve from that standpoint. And that's essentially what we did is we created this partnership with TriLink in order to make mRNAs at a clinical grade level, and sorry, at a preclinical grade level, so that our investigators could work on it.
Mhm.
And really, there's sort of two hopes here. One is that since we're working with TriLink and we're using their SOPs, that any molecule that gets developed in a laboratory would be easily moved to a GMP type production facility. It wouldn't be difficult to move that beyond the preclinical stage. But the other side of it, again, is this idea that if we have mRNAs that are at a reduced cost, it allows the investigators to play with it. It allows us to, try different structures, try different modifications, try different, arrangements of open reading frames, whatever it is, in order to improve upon whatever disease it is that they're after going after. Because, one of the clear ramifications of what's happened in the last 5 years is that mRNA-based approaches work brilliantly for vaccines.
Mhm.
But to get them to that next step, that promise of, cancer neoantigen therapy or gene replacement of monogenic disorders, we really have to innovate in ways to go beyond the biology that is naturally limiting to the mRNA expression space. So in a nutshell, that's really where we're trying to go and why we formed this center. The other aspect of that is, which I think is a very key piece of the RNA Innovation Center, is that we are situated right next to individuals that are world expert in LNP delivery.
Mhm.
Because it doesn't matter how much you innovate in the mRNA space. If you can't get it to the right cells, well, then you're just not going to, be able to do anything. So by having our investigators in the INBT, the Institute for Nanobiotechnology, it really allows us to play these two technologies off of each other and then go after hard to solve problems.
Michelle, how did you end up joining Jeff here at the RNA Innovation Institute? I mean, was it how did this partnership arise?
Quite serendipitously. I
As the
was
best
at
partnerships
PacBio. Yeah,
do. Yeah.
I was at PacBio and I was, well, I should back up a little bit. I was at a startup that rolled out of Hopkins, which was acquired by PacBio. So I really love the startup environment. And I really love PacBio. It was a great company, but I was searching for something more akin to a startup environment. I reached out to Jeff because I saw that he is active in the startup space, and we got to talking. And he said, actually, I have this other thing that's going on, and you might be interested in that. So,
This other
yeah.
thing.
When was that, Michelle?
Guess that was in the beginning of 24, maybe around
Okay.
January that I reached out to you.
Mhm.
Yeah, I think
Yeah.
it was about January 2024.
Nice.
hmm
So what did it look like to you, Michelle, when Jeff said, "Well, I have this thing"? What was the thing at that point? Like, was the thing an idea? Were there bricks and mortar behind it? Was there,
Oh, no, sorry. Yeah, no, I think it was pretty clear even at that point that it was going to be the RNA Innovation Center and what the goal was and what the drive was and the vision for how it would grow and what it would become in the future. Right. I think
Mhm.
that was already in place. So
Yeah.
I was very excited because I was trained in grad school as an RNA biologist, and going into my postdoc, I took RNA into a translational research space. And then when I went to the startup, I took a step away. So I always wanted to come back. So this just seemed like an amazing segue to go back to the place I wanted to be. So
Yeah. Very serendipitous.
Yeah.
Mhm.
What was your charge, Michelle? What did Jeff say? All right, like we're gonna do this thing, and I want you to be on board. Here's what I'd like you to do.
Do you get to make it up as you go
What are you
along?
doing, Michelle? What are you doing?
What do you do, Michelle?
Jeff's listening with very piqued interest.
I
Now.
know.
What are you doing, Michelle?
Yeah, I'm gonna start taking notes here.
Who did I hire, and what has she been doing?
Well, I like to think of our arrangement, and Jeff, you should correct me if I'm off target. Like Jeff is like the CEO, right, and the CSO, and I'm like the COO, right? And I think our visions are aligned on what we want to do here, and I try to make it happen in the broadest sense of that's how I describe it.
Yeah.
Absolutely.
What would you consider the mission of the center like at this point? You've got the TriLink Association, you've got the infrastructure in place to pull things together, and we'll talk about more specifically about funding here in a minute. But as you formed up, Jeff, how would you sort of crystallize the mission of the center now?
Yeah, I mean, the mission really is to try again in an academic, it's really sort of a quasi-academic industrial relationship. And I think that's a very critical piece, especially when you have a technology that really is in its infancy, where you're trying to move it into these new areas. And so its mission is to do just that. It's to take on challenging problems and work with investigators who have expertise in, whether it's cancer biology or some sort of monogenic disorder, to work with them to try and solve a problem. So having, experts in RNA biology, experts in manufacturing, and then experts in delivery, and of course, experts associated with whatever indication, to put those pieces together in a way that is, beneficial and can go through experimental reiteration so that we can learn from each other and then gather that information and then hopefully make inroads into this technology. And I really think this sort of model is the only place this is going to really occur, because by the time something gets spun out at the biotech level, they're already set on their way, right? They have whatever product it is that they're thinking about, their indication, they're going down there. There's not much time to go through experimental iterations. And in the academic setting, in the pure academic setting, there's not necessarily the roadmap in how to commercialize and go after therapeutic development. It's really this, it's always been known as this valley of death between academics and industry. And I think what our center does is sort of fill that void in a unique fashion. So our mission is really that, to put into the hands of our investigators the power to be able to try different therapeutic applications and test them, and then hopefully spin those out, either to market them to other partners or to make their own companies associated with Johns Hopkins. So and a long-term aspirational vision that we have is to make our own, to start from ground zero, some sort of therapeutic, and take it all the way to a clinical trial at Johns Hopkins and have this sort of wholly owned program that we've developed. And hopefully that would take advantage of some of the novel science that we intend to explore.
Mm-hmm.
Yeah, I'd like to add to that too. I mean, Hopkins has a very rich history of taking research from basic science to a therapeutic. And so we would like to be a part of that narrative. And we're hoping to bridge the place where people are at the point of discovery, which, as Jeff said, is more challenging to do if you're in a biotech or a pharma setting. So we can meet people where they're exploring things, exploring ideas, and help them get to the point where they're ready to try something larger in a clinical trial.
Yeah. It begs the question. I'll let you. I know you got one, but it begs the question just for a level set of understanding. You talked about these people, these discoverers, these investigators. Who are these people? Who's invited to come play at the RNA Innovation Center?
All are welcome.
Matt will be there tomorrow at 8 o'clock.
I mean, right now we've started out pretty small, meaning mainly Hopkins researchers, but that's not really small. I mean, we're a pretty big institution. Once we get our sort of the wind beneath our wings, we'll expand from that. But as you know, as Michelle said, all are welcome.
He seemed a little anxious when he
Yeah.
said that, didn't he?
Yeah.
Because
I mean,
he was looking at us in a strange way. Did you see that?
yeah, he's recognizing the power of this platform, and he realizes that when this episode drops, the floodgates will likely open.
Well, to be honest with you, the reason I say that is because I have been working on mRNA biology since 1993, for a very long time, and up until 2019, nobody cared about any of that. It was very esoteric and stuck in academia. And
Yeah.
I've seen sort of how my life has changed since 2019 and the explosion of interest into the research we do into old things that we had done in the past. And it's a floodgate has opened since 2019, 2020 of people wanting to get into this game. Because when it comes to sort of therapeutic development, as you guys know, you've been doing this for a long time with this podcast, therapeutic development is really very particular, right? You have an indication, somebody discovers a small molecule or interesting gene therapy, whether that's base editors or CRISPR or something. But in the mRNA space, it's so easy to really think about things that you might want to try, whether that's creating antigens or whether that's, gene therapies as an mRNA or base editors or, CRISPR as an mRNA. It's just really easy for people to imagine things they'd want to do. And so that does open this floodgate of like, well, hey, I could try my favorite thing now. All I need to do is have some tools on how to do it. And that's the piece right now that a lot of investigators are missing, but they're eager to get involved. Even before we officially launched this, the rumor mill in Hopkins was brewing, and we were getting so much interest of people just coming up and asking, could we do this? And my lab, in fact, started making RNAs for people just on the bench because there was too many people asking us to do this.
Yeah.
So when I say that we start out small, we have to, because I just think we will get flooded
Yeah.
with a lot of requests.
Yeah. Jeff says he's been working on RNA since 1993. I'm like, what? Like you were coloring RNAs with your crayons? Dude, you don't look old enough to have been doing anything since 1993.
It's very kind of you, but it is a fact.
Well, I think, you've talked a little bit about sort of the state of the mRNA space, what it was prior to the pandemic and now where we are today, right? But I think one of the biggest questions I had, when hearing about the center is why now? Right. And so I think every good origin story, right, has a why now. And you and Michelle and Jeff and you and I, we met just a couple of weeks ago, right, at the Alliance for mRNA Medicine's first Ascend conference, right? And so we were kind of privy to a lot of the ongoing challenges that the space is having. But I'm curious, how would you answer the why now question? Why do you think that this center is so extremely necessary today, given what's going on in the mRNA space, and what are some of the biggest innovations that really you think need to be made in this space that you think the center will really help pave the way towards? So maybe we can start with Michelle and jump to Jeff.
Sure.
Yeah.
Yeah, I mean, I think this also answers who we're trying to serve. Like we are starting with the Hopkins community because, as Jeff said, there are a lot of researchers here who are interested in working in mRNA or with RNA generally, but they don't necessarily have exposure or molecular biology skill set in their labs. So we want to bridge that divide. I think it's a time where a lot of people can start thinking about this new modality. It might spark some different novel approaches to treating things that previously were not treatable. So I'm so sorry. I just lost track of your question.
No, what's the origin, why now? Why do you think this is
Why now?
an important time for the center and, given this current state of the mRNA space?
Yes, and I think as following up on what Jeff said, because of the pandemic, because of the efficacy and the clearly proven value of using mRNA medicines, a lot of people want to get into this research area. They don't have the skills necessarily, but we do. We can offer them guidance, we can offer them resources, and we can offer them a pathway to taking their ideas from discovery stage to the point where they might be ready to go to a larger scale study.
Mm-hmm.
Yeah, and I'll just, dovetail on that is the when it there's really two mRNA based therapies that are out there. And they really do represent some of the most widely understood and tested therapeutics that have ever come out. But they're based on sort of one set of rules in terms of optimization of those. And when I mean the manufacturing is one aspect, but then the biology is another aspect. And how many other things are out there that we haven't tried yet, right? I mean, they work really well for vaccines, but mRNA as a drug is a good drug because it's cleared by the body. It's cleared by the body very rapidly. And that's perfect in some applications like a vaccine. It's not great if you're trying to do something like a gene replacement or protein replacement therapy. And then, we're learning other things as we move along. I mean, the pandemic fueled a need to get these vaccines out right away. And so both BioNTech, Pfizer, and Moderna linked, decided to use, M1 pseudouridine as their modification. Well, that might not have been the best choice now in retrospect, because we're getting data coming out in the publication sphere that, those modifications, well, allows for the vaccines to be useful. There could be, unintended read-through events that, we might not want. And there's over 150 unique nucleotide modifications that we know of. Have we explored that entire space? Could we get to a point where we could improve the expression profile of an mRNA and reduce its immunogenicity better than what we already have? And there's real examples where we might need that. So, for example, it's not a case. It's not what we've heard from our colleagues in the government is that the influenza outbreak is not a when, or sorry, if it's a when, that there will be eventually a large scale influenza outbreak, and we're going to need viable vaccines for that. And in those cases, you often have to mix multiple mRNAs together, but you get to a point where you have too much mRNA per dose. And so that creates an immunogenic response. And so can we explore technologies, modifications, or alterations in the mRNA structure that can reduce the overall amount of mRNA needed to get the same amount of protein product, and then allows for us to go into these more complex therapeutic options? And so I think why now? I mean, that was your question. Why now? Is because we don't have, this technology is powerful. We need it. We're going to need it for rapid responses to pandemics. We're going to need it for other, rare diseases that we might want to go after, or just simply reducing the costs of already existing therapeutics because it's cheap relative to other therapeutics. But the why now is because we don't have the decades of knowledge and how to improve upon the efficacy in vivo, but also the manufacturing. And you could even see from the event you were talking about, the Alliance for mRNA Medicines event, there's so much interest in just the manufacturing alone and how we can reduce the cost and make things better and safer. So it really is an important moment.
And understanding too, I think during that conference, there was a really good quote about how we know. So we've done decades and decades of research into protein structure, right? But there's still very little understanding of RNA structure in and of itself. And
That's
so,
right.
a lot of the work that you guys are really well suited to address, right, is to help us sort of answer the questions that maybe biotechs today are not quite capable of answering yet, or because of different risk tolerance, right, and regulatory tolerance there can be some limitations there commercially that aren't necessarily
Absolutely.
faced with academic research.